in a club?
It's been a year and a half, I think. I mean, for me, but there's people who are in the club more than me.
Hmm.
Whereas I think our club is 4 years old. Yeah, 4 or 5 years old
That's
now.
exciting. And what do the students usually do when they graduate?
Most of these students are from medical school, but
Hmm.
I'm biomedical engineering student, for example,
Okay.
and there's some other students are from biology and I think a few from chemistry. I'm not sure. Yeah, but most of them are doctors, like MDs.
Like they're heading. So how does it work there? Do you, like in the US or in Canada, you do an undergraduate degree and then you go on to medical school. Do you enter medical school right from the beginning?
I think it's different. Um, Maybe they can explain it better than me because I'm an engineering student. But
Yeah. Okay.
Well, hello, Karla Kaun. I study medicine, and in Turkey, we directly just go to a medical school. There is no
uh,
undergraduate education. Like we
mhm.
do not study biochemistry or anything else.
There's no
Okay.
pre-med, as I know.
Okay. Well, that helps and makes sense. Thank you for sharing.
Mhm.
And also, how was your sabbatical?
Oh, so far so good. I've actually been spending the first month of it writing grants.
Oh,
So the way that science works in the US is it's usually funded by the government, but you have to
mhm.
write grants to the government. And so this is my rotation where I'm writing a whole bunch of grants to be able to fund my laboratory. Um, it's just sort of like typical what we do, but I just finished that, so I'm going to start on my sabbatical project starting next week, actually. I'm planning to write a book about addiction for my sabbatical.
Oh, that's a really nice plan. I love it.
There's lots of books out there, but some of the books they don't really talk about more recent work in addiction, and they certainly don't talk about the molecular mechanisms of it. So I'm going to focus a little bit more on that. And part of the motivation for that is, some people feel like addiction is in their genes and they don't have any control over that because, other people in their family have had alcohol and substance use disorders and there was no way out. But all the current research, at least in animal models, suggests that we are actually able to change gene expression in our brain. And so that's the type of thing that I want to write about in the book in the context of addiction.
I can't
I really
read
want
it.
to read your books.
Yeah, I can't wait.
Well, it certainly won't be you won't see it for a couple more years. So
Oh.
No, we can't wait, but we are
yeah, no, I think it'll be worth it in the end. It's a bit of a work.
because
Um,
it's a really unique approach, I think,
Yeah,
to the
yeah,
addiction.
I think so too. And the fact that we study flies in a lot of ways, it forces us to understand levels of addiction on many different levels, all the way from humans through primate models and rodent models, and all the way to invertebrates. And because I have this sort of broader understanding of how the questions are being asked, I think it gives me a unique perspective to understand these questions. So we'll see how it goes. Is the audio okay?
yeah,
I can plug my
it's
microphone. No, it's
it's
okay. Perfect. Let me close all the other apps that I have open. Okay. And you said you're going to record this and post it on your YouTube channel?
it'll be live, I think, right?
Oh, live. Okay, so it's being streamed.
Yeah, I think so. Yeah,
Mhm. That's exciting. Just let me know when you want me to share my screen again.
sure. First, I'll introduce you briefly, and then I think, yeah, I'll tell you when to start, then you can just share your screen.
Sounds great. It's not often I get the opportunity to speak to people in Turkey. My
Actually, I'm so sorry, but in the student clubs in Turkey, there are not many teachers who are from abroad, but we want to make something different with this project. We have many teachers from Harvard, Stanford, like many universities.
That's wonderful.
We just want to provide this opportunity to students in Turkey because there are many brilliant minds, but they don't have this opportunity. So that just
My
or
daughter, my daughter's actually learning Turkish right now. Um,
Really?
oh,
yeah, so
that's interesting because it's
That's
not very
surprising.
common.
my spouse is Azeri, and so they don't have Azeri like as a language that you can learn necessarily on any of these language apps. So she's learning Turkish because it's the closest thing so she can speak with her grandparents.
That's really nice. I think that's really a nice idea.
Yeah. So Nejesan, everybody.
Also, if there's anything we can do, we can help. If there's anything we can do, um, I don't know how can we help, but if you need any resource to learn Turkish or anything, we are ready to help. We will be delighted.
Oh, well, thank you so much. I appreciate that offer.
We will be
So
delighted.
far, she uses Duolingo. So if you have other recommendations, I'm all ears.
And someday, if you want to practice Turkish in Turkey, we would love to meet with you in person. Definitely would like to host you in Ankara in our school.
Well, I'll let you know if I'm ever in the area. This year, at least this year, next year, I'm trying not to travel too much so I can get my projects done. Before introductions on Zoom is always so awkward. In my classroom, I started playing music to
Background.
entertain students over Zoom. And then they would keep this Spotify playlist that was just for the classroom. And they would add these songs to try and shock me. It was kind of fun.
What kind of music did you share?
Uh, it was all kinds of music that they added, all the way from, I don't know, like 1920s blues and swing to like really hardcore rap.
I really love blues and I just really love blues.
Yeah, I like that. I think their plan was just to see if they could surprise me. And some of them are actually found themed music, like music that would match the lecture. I was always very impressed that they were able to do that.
They seem really creative students, I think.
Brown students are a bit like
Yeah.
that, the students I teach, yeah. How many students are part of the club typically?
I guess I'm not sure. Ipek, I think you can answer that.
I haven't counted yet, but there are
We're
many students from different faculties and
yeah,
different departments.
very
So
crowded and multidisciplinary.
Mhm.
Yeah.
That's nice. It's wonderful that you have a student-driven club like this. It's a nice way to get to know science outside of Turkey.
Yeah.
um, how are student clubs in Brown University?
They have so many different types of student clubs. There's like a club for every interest and every identification. It's pretty impressive. Yeah. Brown students typically join, I don't know, two or three clubs on top of all of their classrooms and classworks, and they do lots of volunteering and these kinds of things, especially those students that are interested in medical school.
Right. Well, what sort of events are they doing? Like, for example, recently we had a congress, multidisciplinary neuroscience congress, and it was pretty crowded, and I think it was pretty successful. And do they do such things like that?
That's really interesting. So typically it's not necessarily organized around like an academic topic, but I was recently, for example, they had a conference for women that were in STEM fields.
Mm-hmm.
And so I was one of the panelists, and there was a lot of other sort of professors there, and they had a bunch of different types of events to really focus on, promoting women in science. And I thought that went pretty well. It's pretty impressive. Um, and a lot of the topically organized things at Brown are sort of organized by the administration or by faculty. And in the neurosciences, there's tons of different opportunities because there's the Carney Institute for Brain Science. And this brings together, I don't know, hundreds of faculty from like 30 different departments. So there's lots of people at Brown that aren't necessarily in the neuroscience department that are doing neuroscience. They might be doing molecular neuroscience or engineering or philosophy or public health or medicine. But they're all doing sort of like science focused on understanding the brain and behavior. And so it's kind of a unique place to do this kind of work. And every year they organize a number of different symposia, and all of the undergraduates and graduate students are invited to attend. So they do things like that.
So I think we're good to go. Ladies and gentlemen, good evening, and Dr. Kaun, good morning. I'm truly honored and excited to welcome you all and want to thank you for attending our event. And we'd like to express our gratitude to Dr. Karla Kaun for being here with us tonight. As Ankara University Neuroscience Society, we are very excited, flattered, and happy to host a distinguished scientist as you in here. So before the presentation, I'd like to introduce Dr. Karla Kaun briefly. Karla Kaun is an associate professor in the Department of Neuroscience at Brown University. She's a behavioral neurogeneticist fascinated with the genetic, molecular, and cellular mechanisms of memory and addiction. She received a bachelor's degree in psychology from the University of British Columbia, where she worked with Dr. Catherine Rankin, and a PhD in zoology from the University of Toronto, where she worked with Dr. Mala Sokolowski, and completed her postdoctoral work at the University of California and Howard Hughes Medical Institute, where she worked with Dr. Ulrich Heberlein. Her team investigates how alcohol and drugs of abuse influence the molecular mechanisms underlying memory formation to induce cravings by combining behavior with in vivo imaging, genetics, molecular biology, biochemistry, and bioinformatics in Drosophila melanogaster, which is a type of fruit fly. Karla is currently president of the International Behavioral and Neural Genetics Society and an associate editor of the Genetic Society of America peer-reviewed journal, Genetics. She is the 2023 recipient of the National Association of Biology Teachers Genetic Education Award, loves collaborating, and is an advocate for innovative teaching methods to decolonize STEM. I strongly encourage you to use this opportunity to ask Dr. Karla questions by filling out the form provided. So without any further, I'll leave the word Dr. Karla.
Thank you for that really wonderful introduction. Here, let me share my screen here. Okay, are you all seeing this okay? Maybe this is a little bit better.
It's all fine.
Okay, great. If the audio or if the visuals get disturbed at any time, please let me know because I don't always see this. And it's a wonderful opportunity to be able to chat with you all today. I'd like to make this as informal as possible, so please feel free to interrupt me if you have questions. And at the end, I'm happy to answer questions about science, but also about life and what it's like being a scientist. And I'll tell you a little bit about where I'm from, and then I'll tell you a little bit about what I do. So I grew up in a remote area in northern Saskatchewan, which is an area in the middle of Canada. And I grew up in a Native American community. So I had kind of an interesting upbringing as a typical scientist, perhaps, but I think it provided me with a really unique way to think about the types of science that I study. And what I study are really the in-depth neural and molecular mechanisms underlying alcohol and substance use disorders. And so I'm really interested in how alcohol and drugs affect our brain and in finding sort of new and innovative ways to help find a solution for alcohol and substance use disorders. And all of the work that we do is very fundamental neuroscience. And so you won't see any of the stuff that my lab works on necessarily leading to a treatment in the next few years. But what I feel it's doing is really revealing some of the fundamental principles of how our brain works and how drugs and alcohol influence memory. And you might ask, why is memory so important in this context? And I'll talk a little bit about that in the future. Now, before I begin, I really want to acknowledge that all of the work that was done that I'll be talking about today was done on the traditional homelands of the Narragansett people, which live in Rhode Island today. And it was done by this incredible team of people. And so one of the things that I love most about science is that you get to work with incredible scientists and collaborate readily. And the circuits work that I'm going to talk about today was done by Dr. Kristen Skaplin, who's now an assistant professor at Bryant University, which is also in Rhode Island. Dr. John Hernandez, who's currently completing his postdoctoral work in the lab. Dr. Emily Petruccelli, who is now an assistant professor at Southern Illinois University, Edwardsville, which is sort of in the middle of the United States. Tariq Brown, who is a senior graduate student in my lab, and Amanda Waterman, who's a technician in the laboratory. And so most of the work that I'm going to talk about today is focused on alcohol use and abuse. And you might wonder, why are we studying alcohol as opposed to other drugs? Aren't other drugs the ones that are really causing a lot of the problems in society? And in fact, alcohol is one of the top causes of death worldwide. And this is not only for its direct role in things like vehicle accidents and suicide and overdose, but it's also through domestic violence and the compounding effects of how even low doses of alcohol can affect the cardiovascular, the respiratory, the digestive, the immune, and the nervous systems. And so alcohol use disorder, like all addictions, it's a relapsing disease. And so our current treatments for alcohol use disorder, including things like naltrexone, they can reduce cravings in some people, but they're really not very effective for a lot of other people. So how do we address this? And part of the problem is that we really don't have any idea of what it is going on in the brain that's driving these behavioral patterns. And so we really have to understand how cravings happen at a molecular level before we're able to, I think, better treat them. And you think, okay, so are there differences in the brain in people with alcohol use disorder? And most of the research today suggests that there are, right? So the brains of people with alcohol use disorder work differently than the brains of people without them. And we're starting to get a handle on some of the neural circuits underlying this. But what we found so far about the molecular mechanisms underlying this is that it's incredibly complex. There are receptors along the cell membrane, ones that might be channels or G protein coupled receptors that change cell signaling mechanisms inside of the cell. There's also other receptors that signal directly to the nucleus and then can change gene and protein expression inside of the cell. And there are also direct changes to the way that chromatin is wound inside of the cell and the way that genes are turned on or off. And the incredible complexity of this is that it works different in every different cell type. Right? So how on earth do we get a handle on this? And to sort of put this in perspective, in our brains, there are about 100 billion neurons, right? And each of our neurons is connected to up to, say, 10,000 other neurons. There are maybe 100 to 400 billion stars in the Milky Way, right? And so that's an incredibly complex problem. If you're having 10,000 connections for every single star in the Milky Way, how do you possibly get a hold of this, especially if the cell types are really as heterogeneous, or there's so many different types than we think right now? And there aren't a lot of good models to really understand this at this level. In mouse models, for example, we're just discovering how incredibly complex neuronal anatomy is. So this is a mouse brain looked at from the top down. And this is an example of only 302 neurons inside of this brain, right? And that already looks like an incredibly complex sort of, I don't know, spaghetti ball of neural processes. And to put this in context, that's only 302 neurons, but in the mouse brain, there are about 71 million neurons. So you can imagine the levels of complexity in neuroanatomy and gene expression or protein expression within each of these individual cell types that needs to be understood before we understand how we're able to figure out how to change molecules that can drive behavior in the brain. And that's why my lab studies the fruit fly, Drosophila melanogaster. So this incredibly cute little creature here is the fruit fly. And this colorful structure inside of its head is its brain. And we know a lot about the fruit fly brain. We know all of the major structures of the brain. In fact, we have a connectome of at least 2/3 of the brain, and that's growing on a daily basis due to community efforts. And we have genetic tools that allow us to basically precisely map and manipulate almost every neuron that we want inside of the brain. And this is true for the area of the brain that I study, which is the memory structure in the fly brain. And so this L-shaped structure here is what we call the mushroom body. And we have a really good understanding now of how this structure works in the context of memory. And we have genetic tools that allow us to basically manipulate almost any neuron we want in that circuit. And so what this allows us to really do is to look at individual genes and proteins inside of this circuit, or manipulate each of these individual neurons inside of this circuit and test that in the context of behavior. So it's a model that can provide exactly what we need to understand how cravings are formed, basically. And you might ask, okay, well, fruit flies are slightly different than humans, right? There's 500 million years of divergent evolution. But what's really, really interesting about flies and primates like us, for example, is that both flies and hominids experience alcohol in fermenting fruit in their natural environment, right? So our brains are actually much more related to how a fruit fly's brains are in this context than a mouse brain, for example. And this can explain why primates and humans such as us like alcohol sort of off the bat in a way that fruit flies do. And so not only do we experience alcohol in our natural environment in similar ways, but the circuits underlying how reward is sensed and how it's remembered is remarkably similar between flies, rodents, and primates, right? And in this context, we didn't understand this until about 15, 20 years ago, I would say. We used to think that flies have this neurotransmitter called dopamine, which is kind of like norepinephrine, that mediates reward. And what we understand now is that, like us, it's really dopamine that's at the root of how reward is formed. And it turns out that dopamine modulates dopamine in a very similar way to the way that norepinephrine modulates dopamine in our brains. And so we have a good understanding now of sort of the circuits mediating this. Do flies actually act similarly to humans in the way that alcohol affects their behavior and brains? And so the way that we do this, that test this, is we give flies volatilized alcohol. And you might think, okay, this is kind of strange. Why would you volatilize the alcohol and get the flies to breathe it in? And the reason we do this is because it's much easier to control the dose of alcohol. And in fact, humans get intoxicated this way too. For example, you can buy something called a vaportini, which is basically an alcohol bong. It has a tea light in the bottom, and then they put a martini in the top of this, and then they volatilize the alcohol and people breathe it in. And it's marketed as a way to imbibe alcohol without the calories. Now, I don't recommend this device at all, just so you know. It does make one intoxicated, but it's really, really bad for the lungs. And so I wouldn't recommend it. In any case, though, we give flies these intoxicating doses of alcohol, and then we look at their behavior by tracking it. And it turns out with low doses, flies do show behaviors fairly similar to us. They increase their locomotion, and they become disinhibited. So they bump into each other a lot more. The courtship behavior increases. Male and female flies start to court indiscriminately, and male flies become more aggressive with fairly low doses of alcohol. With higher doses of alcohol, flies aren't able to basically walk in straight lines anymore. They have trouble walking up a vial, which is what flies typically like to do. And with even higher doses of alcohol, flies do behaviors fairly similar to humans, and that is they basically pass out in the bottom of the vial. And what I find is really, really remarkable is that the doses similar to make flies sedate are really similar to the doses that's required to make humans sedate once you control for body size and body mass and these kinds of things. And so I think that tells us that there's something very highly conserved with how this simple molecule, ethanol, affects all brain systems. And that makes flies a particularly good model to understand what alcohol is doing to neurons in the brain. And you might say, okay, so flies can get intoxicated, but can they really form cravings? And so what are cravings, right? So imagine yourself going to your favorite bar. And in front of your bar, you order your favorite beverage. And then you reach out to your beverage and you feel the coolness and the smoothness of the glass. And you pick it up and you move it towards your face. And you smell the bouquet of the beverage. And you tip the glass to your lips. And you taste the bitterness and the sweetness of the liquid within. And so all of these sensory experiences, maybe it's the dim lighting of the room. Maybe it's the feel of the glass of your hand. Maybe it's the color of the liquid in the glass. Maybe it's the smell of the liquid itself or the taste. These are sensory cues that become associated with the feeling of intoxication. And it's these sensory cues that are thought to be really important in eliciting cravings. And so when I started my postdoc in 2007, this was the question that I wanted to ask. Can flies form these sensory memories for intoxication? And the way that we study this is we give flies an odor the same time that they're getting volatilized doses of alcohol. And we ask if they're able to form an association between the feeling of being intoxicated, that is the pharmacological properties of alcohol, and the odor that we give the flies themselves. And the way that we do this is we use a typical sort of memory paradigm in flies. We give them one odor that isn't associated with alcohol, and then we give them another odor in which the alcohol is gets associated with this other odor. And then we give them a 1 hour break. And then we give them another training session like this. And we give them another hour break and another training session like this. And then we ask the flies, do you prefer after they sleep it off 24 hours later, do you prefer to go to the odor that was previously associated with alcohol? Or do you prefer to go to the odor that wasn't previously associated with alcohol? And if flies liked the feeling of being intoxicated, they would go towards the associated odor. And so when we count up all of our flies, we actually find that flies indeed do like to go towards the odor previously associated with alcohol. Now, in order to be really rigorous about this and to make sure that the flies weren't just going towards an odor that they liked, we flipped the odors around so that the first odor was associated with alcohol instead of the second odor. And then we asked flies, do they prefer to go to that odor? And indeed they do. And so we know that it's the association between any odor and the feeling of it being intoxicated that is driving the learned preference. And so what's really remarkable about this particular memory is that it lasts a really long time, up to 7 days in flies. And that's a pretty long lasting memory. Flies don't live all that long. And so it's really remarkable that they show memories at this stage. And what's even more remarkable is that the flies will walk over 120-volt electric shock to get to the odor that was previously paired with alcohol. Now, they won't walk over that strong of an electric shock to get to an odor previously associated with sugar, which is a more natural reward for flies. And so we know that there's something intrinsically very interesting about these types of memories, and we want to understand how that's working on both the neural circuit level and the molecular level. And to study this, what we do is we focus on one particular reward circuit in flies, and this is the reward memory circuit. And this is the structure that I showed you earlier, this L-shaped structure. And this is what it looks like. There's about 2,000 cell bodies in this structure. And when a fly is presented with an odor, that odor is sparsely encoded within this structure. So instead of all 2,000 neurons lighting up when the odor is presented, maybe only 10 neurons will activate. And then those neurons extend their axons into this L-shaped structure. And then these axons of these Q neurons here are completely innervated by dopamine in a really beautiful patchwork sort of array. And for every dopamine neuron that goes in, there's a glutamatergic, a GABAergic, or a cholinergic neuron that has its dendrites in the same compartment. And then these neurons extend their axons to other types of neurons. Maybe it's back to the dopamine neurons in a recurrent loop. It's maybe to each other because these neurons are highly connected to each other, or maybe to other areas of the brain. Right. And so the first goal for us was to figure out what alcohol is doing to these dopamine neurons themselves. We want to make sure that alcohol is actually eliciting a rewarding response in the way that we might expect. And so in order to do that, we drove GCAMP7, which is a GFP localized sort of calcium reporter in flies, specifically in these dopamine neurons here. And then we gave flies a regular dose of alcohol while they were walking on a ball, basically. And we looked to see how activity level was changing as the level of alcohol increased in the flies. And what we found was that at first, the smell of alcohol, we presume, was reducing activity of these dopamine neurons. But over a 10-minute period, the presentation of alcohol was actually increasing activity of these neurons compared to what you would typically see with just an odor. And that told us that, in fact, alcohol is having a massive effect on these types of neurons. We tried to localize this to a particular subset of neurons, right? And what we found was that only a small subset of dopamine neurons were important for remembering that. So it looks like when you make a memory, you need activity of this entire subset of neurons. But when you remember the memory, that is, the flies are presented with the cue, and then they go towards the cue previously paired with alcohol, only a small subset of these dopamine neurons were involved. And we performed a neuronal behavioral screen to figure out what are all of the different output neurons that were important in that memory. And we found only a very small subsection, glutamatergic neurons that were connected to this one small subset of dopamine neurons, and a cholinergic neuron here that was also connected to these dopaminergic and glutamatergic circuit here. So what's interesting about this particular circuit itself is that these green neurons here are thought to encode an aversive signal, and these blue neurons here are thought to encode an appetitive signal. And so why that's particularly interesting for us is that alcohol, like all drugs of abuse, have both appetitive and aversive properties. The brain at the same time. And so this is a perfect circuit to understand how the rewarding properties override the aversive properties. Like, why do people keep going back to alcohol if it tastes bad? Because we have a connection now between the appetitive and aversive properties of alcohol. And we wanted to find out where these two different circuits merge. And so we used a tool by our collaborator, Gilad Barnia at Brown University called TransTango. And we mapped all the postsynaptic connections to these three neurons. And what we found was convergence in one particular area called the lateral accessory lobe here. Yeah. So this is a structure right behind the structure that's important for encoding fly smell. And we wanted to ask if manipulating these particular neurons would end up eliciting a behavioral response. Because the hypothesis is that these appetitive and aversive circuits merge. And then at that merging point, a behavior to either go towards the odor or to avoid the odor is initiated. And so we activated this particular subset of neurons that we mapped, and we found that indeed it increases how much the flies will move and the directions that the flies will move. And so that provides a really, amazing area to be able to understand where the behavioral choice is being made. And so the question that we're trying to understand now is, how memories are encoded and how they're remembered. And then how do we go from remembering the memory to initiating a behavioral choice? And what we're really interested is understanding is, these circuits are not the same circuits. So what changes between making a memory and remembering a memory and initiating a behavioral choice? There have to be some molecular changes happening here because it's not the same circuits that are required for both responses. And so a good chunk of my lab is really understanding what are the molecular changes that correspond with this circuit plasticity. So which genes are getting turned on or off? How is chromatin being modified? And in order to understand that, we first did a large-scale genetic screen. So we screened through hundreds of different mutants that we know had expression in this memory structure of the fly brain. And we found a very interesting signaling pathway. And this signaling pathway is called Notch. And so what is Notch, you might ask? So Notch is a really well-characterized cell-cell signaling pathway in the context of neuronal development. And what it is you have this receptor that sort of sits on the cell membrane. And then in the cell next to it, you have the ligand for that receptor. And when the ligand gets close enough to the receptor at the cell membrane, there's this gene called scabrous, which is thought to scaffold the ligand to the receptor. This gene scabrous is the one that we pulled out of our genetic screen. And so when these two things combine and scabrous helps hold the ligand to the receptor, the intracellular domain of the Notch receptor gets cleaved. It gets transported to the nucleus, and there it binds to its cofactor, which in flies is called suppressor. In long-term memory. And you think, okay, well, all you pulled was really something that's important for long-term memory. That isn't interesting. But it is, because what we're trying to understand is how alcohol messes with these long-lasting memories that are important for inducing cravings, right? The memory for a sensory experience can last someone's entire life. And then they'll be, recovered from an alcohol or substance use disorder. 20 years later, they'll be, by chance exposed to a cue and they get a craving. And so if we can really understand sort of that process of how these types of sensory memories are made, and how alcohol messes with them, then maybe we'll have an idea on how to better treat these cravings. Because Notch had recently been implicated specifically in development, or how it had previously been implicated in development, we wanted to make sure that we weren't just showing some kind of morphology defect in our mutants. And so what we did was we knocked down scabrous, notch, or suppressive hairless, specifically in adult mushroom body neurons. And we found that this prevented memory formation. And so that told us that indeed, this is an adult phenotype. This is an adult phenotype specific to memory formation. And it's important, and it's required for the canonical signaling. So some kind of signaling that was actually affecting gene transcription. And we were very excited by this because we wanted to know if alcohol was affecting gene transcription directly through this pathway. And so what we did was we gave flies alcohol, and then we used an artificial notch reporter developed by Toby Lieber. And so what this receptor is it's sort of like sitting on the cell membrane, and instead of a normal intracellular domain, it has a Lexay domain. And when alcohol sort of hits this, or when this ligand binds to the receptor, the Lexay domain gets cleaved, transported to the nucleus, and it drives expression of GFP, a destabilized GFP. And so basically, we just give flies our normal amount of alcohol, and then 24 hours later, look to see how many glowing green neurons are in the fly memory structure. And indeed, 24 hours after memory formation, we saw a significantly greater number of notch active neurons in the fly mushroom body. And so naturally, we wanted to figure out, what are these targets? So which gene targets are being affected directly by alcohol? And to do that, we did an experiment called CHIP, where we were looking at all of the sort of chunks of DNA that were still bound to the suppressor of hairless protein. And we started first looking at dopamine 2 receptors, because we noticed that dopamine 2 receptors had a whole bunch of putative binding sites for the suppressor of hairless gene. And so we did experiments where we looked at how alcohol impacts the amount of suppressor hairless bound to D2 receptors. And we found that alcohol actually reduces the amount of D2 receptors, not throughout the entire gene, but just near the promoter region, which is important because that's probably what's driving transcription of this gene. Right? And so we were very excited about this because it gave us the very first evidence that alcohol is messing with the brain's natural memory transcription pathways. And it's doing so through notch signaling, which is, in almost every neuron in the brain, but has special roles in memory neurons. And that's true for flies and mice and rats, and probably humans too, because notch 1, so humans have 4 different notch genes, flies have 1, but the one that is most similar to the fly gene has been implicated in Alzheimer's disease in recent years. And so this might be a pathway through which alcohol can influence this process. So we loved the approach that we took because it showed a functional relevance to changes in gene expression inside of the brain. But it's also a technically very difficult approach because you had to screen through mutants, and that sometimes took years, right? And so to gain a much broader understanding of all of the things that were changing at different time points in the mushroom body, we performed RNA sequencing. And to do that, we tagged the mushroom body nuclei with a worm nuclear membrane protein using a technique called intact. And then we pulled all of those nuclei out and sequenced them to understand how is gene expression changing when these alcohol memories are formed. And we found something rather interesting. And this is work done by my previous postdoc, Emily Petricelli. And what she found when she looked at all of the transcripts that were expressed in the fly mushroom body was that when you form an alcohol memory, certain transcripts go up and other transcripts go down, right? But it's not all transcripts, and it's not all genes either. So when we first looked at changes in gene expression, when you're considering all of the different forms of genes, all of the different transcripts, we didn't find very many differences at all. It wasn't until we looked at individual transcripts that Emily found differences. And what she found was that some of these transcripts would switch from one form to another form. And I'll tell you an example about just one of these. And that is a gene called STAT92E in flies. Okay. So STAT92E in flies is the fly stat. And what STAT is, it's a transcription factor. So it binds to DNA, much like suppressor of hair loss that I had previously talked about. And it gets activated because of this particular signaling pathway called the JAK-STAT signaling pathway. And in this pathway, there's a receptor tyrosine kinase here. Yeah. And it's sitting on the cell membranes of mushroom body neurons, much like Notch was. And when the tyrosine kinase sort of gets activated when a ligand binds to this receptor, the STAT goes into the cytoplasm, it becomes phosphorylated, and then it binds to each other. And when it binds to each other, it gets transported into the nucleus and it binds to DNA to drive transcription. And so very similar to the Notch signaling pathway that I told you about before, the JAK-STAT signaling pathway is a potential mechanism through which alcohol or drugs of abuse can influence gene expression in memory neurons. And so why were we particularly intrigued in this with this pathway? Well, part was for the reason that I just told you. And part was because nobody had really discovered a role for this pathway in this context before. JAK-STAT signaling is largely studied in the context of cancer or immune signaling, right? And we know very little about how it affects neuronal plasticity. We do know that it's expressed in neurons, and that's true in many different types of organisms, but we don't really know what it's doing there. And so the first thing that we wanted to do was ask if this signaling pathway was functionally relevant, right? And this is a very important thing to ask because when you're doing transcriptomic studies, you can find lots of changes in gene expression, but you don't necessarily know if those changes are causing changes in the function of the cells themselves. Flies are a really great model to ask this because you can temporally manipulate gene expression in very specific ways. And so we've reduced expression of this gene just in adult mushroom body neurons, and again, reduced memory expression. And so that told us that it's a functional test. And so what are the differences between these two different genes, you might ask? Well, one of the transcripts is normally expressed, and this is the RI version. And then when you form an alcohol memory or a sugar memory, which is data that we haven't yet published from the lab, we get preferential expression of the RH version. And what are the differences between these two different versions of transcripts? It turns out that the RI version is has a truncated form of the transactivation domain. And this is an important part of the protein that binds to DNA. And so what we hypothesize is that we're getting conversion to a form of the gene that is more transcriptionally active, right? And so that probably affects transcription in Q encoding neurons. We don't know exactly how that happens yet, but my lab is actively studying that. And one of the ways that we're studying that is by making isoform specific mutants for the different STAT versions. So taken together, what is this exactly that we're trying to do, right? We're messing with genes one at a time in fly brains and showing how this affects behavior. How is this significant in a bigger picture question? Well, our neurons, like fly neurons, have all of these sort of JAK-STAT and Notch signaling pathways. They have a lot of other typical pathways. And these pathways are typically studied in the context of neuronal development, right? So Wnt signaling, EGF receptor signaling, and Notch signaling. What these pathways all have in common is that we thought that once your neurons are formed, they basically turn off and they're not really that active again, unless something happens like cancer. And then these pathways can get turned on and overly active. What our data suggests is that these pathways are active in the context of brain activity, and brain activity that's important for memory formation. So that's the first thing that we think that we're finding. And the second is that alcohol, and indeed we have evidence now that other types of drugs of abuse like nicotine and methamphetamine are influencing these pathways in really interesting ways. And one of the ways is it's changing the genes that these pathways will normally turn on and off, right? We know that when we form long-term memories, it's really important to have transcription. This is typically studied through the Krebs signaling pathway. Cyclic AMP and Krebs signaling are known to impact lots of gene expression important for synaptic plasticity. This is just the tip of the iceberg. There are so many other signaling pathways expressed inside of these cells. And our lab's goal is to really understand how some of these signaling pathways act in the context of addiction. And so one of the things that my lab is contributing to the field is really defining molecular mechanisms within circuits in behaving animals and understanding how they act in a natural context, and then to understand how alcohol and drugs of abuse influence this process. So, will this really lead to more information on how cravings are formed? Well, I think that that's a very difficult question to tackle. But we do know that all of these signaling pathways have been discovered in the context of alcohol use disorder in many different types of organisms. The problems in the field right now is that we don't always have the resolution to understand how each of these different molecules are functioning in the context of behaving animal. And what the fly presents us with is the ability to really get down and understand exactly what this protein is doing or what this protein is doing and how that's manipulating gene expression. And then how these changes in gene expression are then in turn affecting activity of the neuron, and then how activity of that particular neuron is influencing behavior of an animal. And so will this actually lead to anything useful in understanding how human alcohol and substance use disorders are formed? Well, I hope so. And the reason why I say that is because now what my lab has shown is that alcohol alters neural dynamics in natural reward memory circuits. So we have definitive evidence to show that. We know that individual variation for alcohol preference is encoded within memory circuitry. And this is a project that's largely spearheaded by John Hernandez in the lab. We know that gene expression in memory circuits is dynamic. And so one of the ways that we know this is because when we did our RNA-seq experiments, we were looking for things that changed just within 24 hours. And like I said, we didn't necessarily find major changes in gene expression in that, like, the entire gene didn't turn on or off. What we found was a bit more subtle than that. We found sort of subtle changes from one form of gene being expressed to another form of the gene being expressed. And these were very dynamic, right? They happened within 24 hours, and they happened as a consequence of alcohol exposure and or memory formation. And so these changes are very dynamic, and we don't understand yet how dynamic these changes are in flies or in other organisms. And I think this is a really interesting question to be able to understand how our activity of the cells influencing alternative splice isoforms. We know that alcohol is directly acting on highly conserved cell signaling pathways, and that it's altering transcription that's important for both memory formation and plasticity of these cells. So, is any of this work applicable to our current understanding of addiction or how addiction is treated? I would say that in some ways it is very relevant because we're starting to understand at a very basic fundamental level how these individual proteins are changing in the context of memory formation. So the types of questions that we're addressing are, how are these memories lasting so long, right? What is changing to result in cravings? And, in the future, can we actually use flies to be able to figure out how to change those cravings back, or how to eliminate just the cravings without impacting other forms of memory? We can't do this yet because the types of drugs that we typically use sort of have many different effects in the brain. And so by using sort of the temporal precision that we have, spatial and temporal precision we have to manipulate gene expression in the fly, we might understand approaches on how to do this. So basically, what this is allowing us to do is put together the building blocks of behavior to understand how psychoactive substances can influence this at a very highly conserved level. And with that, I'd like to thank my lab. And so this is my current lab right now. It's a mix of technicians, postdoctoral fellows, graduate students, and undergraduates. And these are the people that I mentioned before that really helped drive this work forward. All of the work that we do in the lab is dependent on sort of the generosity of people within the fly community. And so the wonderful thing about working with flies and invertebrates is that people are often willing to share really sophisticated genetic tools and help you troubleshoot them. And so these are some of the people that contributed tools and advice that have helped drive the research program forward. One of the other things that I really love about the type of research we do is we get to collaborate very readily. And so we have people that we collaborate on some of the fundamental principles of gene expression, but also how our changes that we're seeing in flies can actually help people with alcohol and substance use disorders. And these are some of the people here. And I'm very grateful to have the opportunity to do this research through the funding agencies. And with that, I'm very happy to take any questions that anybody in the audience has.
Thank you very much for the presentation. It was highly informative. So if you have time now, if there's a question, I'd like to ask the questions. So dear participants, they're waiting for your questions. Again, you can send your questions on the form which is provided during the presentation, I guess. I'm checking it.
Well,
Okay,
Mohammed,
first
thank you very
question.
much.
Okay, the first question. What is the mean addiction? Causa interested in that. Okay.
What's the meaning of addiction?
What is the mean addiction? I'm not sure what they meant. I think they're asking for an definition.
Oh, a definition of addiction.
Mm-hmm.
Yeah, that's actually a surprisingly difficult question to answer. So, addiction is a very complex psychiatric disorder in humans, right? And I would say that humans are probably the only organisms that can really form the type of addiction that the medical field typically describes. And so the DSM-4 and 5 have very specific criteria for having an alcohol or substance use disorder. And one of those, for example, is they have to show tolerance to the drug. Another one of them is they have to demonstrate that they're willing to overcome highly aversive stimuli to seek the drug. And so this is actually one of the hallmarks of addiction, this compulsion towards sort of getting the drug. And this is something that's been very difficult to study in animal models, including flies. It wasn't until fairly recently, I think, that people showed that flies are willing to overcome an aversive stimulus. The work from my lab was the first to show that flies will walk over something aversive like shock to get to alcohol. And there was a recent paper in Nature that was really beautiful from Scott Waddell's lab that showed the mechanism through which this might happen. Because flies typically don't like to walk over shock to get to sugar. But if you activate a very specific subset of dopamine neurons in the fly mushroom body circuit, they will walk over this shock. And part of the reason we understand that is because the reward system and the aversion system in the brain are really tightly connected. This is true in flies, and it's probably also true in us, although we don't know as much about our own neural circuitry. So addiction is actually a very complex phenomenon. But I think, in all countries, one of the ways that people measure it is, are you willing to give up really important things in your life in order to keep seeking your substance of choice? And that's also a very difficult kind of question to ask in animal models, right? Which is why we tend to focus on some of the more simpler questions, like how is a craving formed?
Okay, thank you. And another one, they ask if we can add her on LinkedIn.
If you can add, sorry. Oh,
Yeah, if they ask if we can add her on LinkedIn.
sure.
Okay, so another question. To remember something better, do we need to get a dopamine after receiving that information or before that?
To understand, so the interesting thing about this is dopamine is actually not a very good thing to treat any of these types of memories with. And that's because our dopaminergic systems are extremely complicated. And we know that just, feeding flies L-Dopa and giving people L-Dopa doesn't actually help addiction very much at all. And we know that because it's treatment for people with Parkinson's. And some people with Parkinson's have addictions, just like people with every kind of disorder can potentially have addictions. And so it's much more complex, and it depends on exactly which type of dopamine neuron that you're manipulating. And it's also because dopamine neurons encode both aversion and reward. And we can't just give drugs for these because we don't have good ways to target drugs specifically to the neurons that mediate individual responses in our brains. So just eating drugs usually affects like the entire brain. And that's not yet effective treatments. So that's partly why we're doing the work that we're doing. We know that we can't just mess with dopamine signaling. We know we have to find other molecules that are expressed in those cells that we can target pharmacologically. And flies are a good model because we can target the individual cells to find what those different molecular targets are.
Thank you for your answer. The other one, as I understand, most of the research revealed epigenetic genetic change like decreasing in the D2 receptors, but do you think are the genetic changes caused by chronic alcohol intake that inherited to other generations?
Oh, like transgenerational epigenetic changes. The changes that I talked about today are not those types of changes. These are more immediate changes that are directly affected by alcohol itself. So the question that you asked are, can you pass down these genetic changes to your progeny, or can they be passed down? We don't actually know the answer for that in humans yet. And part of the reason why is that we don't know if we can pass down epigenetic changes transgenerationally in humans yet. There's some evidence that some of these can be happening, like there are populations of people that they studied from World War II that didn't have enough food, and their epigenetic landscape looks different in their children, probably because of this sort of like major stressful experience in their early life. Now, we don't actually know if that's the case in the context of addiction yet. We do know that alcohol can cause epigenetic changes. There's a really beautiful study in mice that actually showed that alcohol consumption can go straight to the brain in some ways and cause epigenetic changes. So what happens was mice were fed alcohol. The alcohol goes to the liver where it's metabolized. And then one of those metabolites from the liver is acetaldehyde. That gets transported up to the brain, and it gets into the neurons in the nucleus accumbens, and it actually binds to histones. Histones are these proteins that have DNA wound around them. And when the histones are when the acetylated, what happens is that they can open up, and then that allows transcription factors to bind and drive gene transcription. So alcohol itself can directly affect the epigenetic landscape. We don't know if this is inherited transgenerationally because we don't know if this can be passed down to gametes. Probably not in that way. But there might be other ways, like microRNAs, for example, which are tiny little genes that we don't necessarily know what a lot of them do yet, but we do know they can suppress gene expression. Those can be passed down to oocytes, and this or sperm or the gametes, right? And so those might be one of the ways that some of these changes can be made. But the direct changes in D2 receptor expression, unlikely.
Thank you. And another is, can you explain the molecular basis of alcohol withdrawal symptoms, such as seizures and anxiety, and how they relate to neural pathways and neurotransmitter systems?
Wow. That's a pretty complicated question. One of the ways that withdrawal, one of the mechanisms through which this withdrawal happens is through channels. Because the seizures that you get in alcohol withdrawal, for example, and alcohol is really interesting because it has some of the worst withdrawal symptoms out of all of the different types of drugs. And withdrawal can cause death, actually, in people that are chronic alcohol seekers. And we know that it directly affects potassium channels, and that can affect neuronal excitability. We also know that alcohol has the potential to bind to GABA receptors, which can sort of reduce inhibition, which then increases excitability. So you have a lot of hyperexcitabilities in lots of different ways. What's interesting about how alcohol binds to these GABA receptors is that it doesn't seem to affect the preference, but it can affect tolerance. Yeah, and it can affect sort of withdrawal through that mechanism. So alcohol can affect, many different types of effects through withdrawal. Now, whether that's related to relapse is unknown. Yeah, relapse is a bit more complicated because it involves cravings as well as withdrawal.
Thank you. And another question. What is known about the relationship between alcohol addiction and impulsivity, decision-making, and emotional regulation from a neuropsychological perspective?
Wow. You all have really, really interesting questions. I can't tell you all that's known because people have written books on this, but I can tell you that in humans, chronic alcohol consumption and chronic drug consumption of all different types of psychoactive substances actually affects impulsivity pathways, right? So it affects many of the neural pathways that are normally important for decision making in the brain, and it increases impulsivity. In human studies, it's a bit difficult to figure out what came first, the chicken or the egg, and that is, did people that were naturally more impulsive seek alcohol or other types of drugs because that was part of their personality? Or is it that alcohol and drugs are actually directly impacting impulsivity? And I think most of the work in animal models suggests that it's indeed both, right? And so we know that dopamine signaling is a very important impulsivity, but we really don't know that much about the molecular mechanisms mediating this. I hypothesize that what's happening in these dopamine pathways, similar to what's happening in the fly brain, is that, changes in activity of these circuits over time because of the alcohol and drugs is changing gene expression. And that is increasing activity in these pathways. So you get disinhibition and increases in activity in impulsive behavior, and then inhibition of the pathways that are important in stopping the impulsive behavior, because there's transcriptional pathways that are turned on in some of these cells and off in other of these cells. We don't know what those pathways are yet. In rodents, we know mTOR signaling is probably important for it. So it's one of the transcriptional translational pathways through which this is happening.
Thank you. Another question. Why does alcohol's effects on our emotions differ time to time, even though the chemical is the same? I mean, sometimes it makes happy, energetic, whereas sometimes increases depression. I don't remember myself getting sad when I eat chocolate.
Mm. Hopefully you'll never eat enough chocolate to make you sad. Um, we don't understand the reason to that. We do know that internal state, and that is like how we feel at the time, is really important in how alcohol is affecting your neurons directly. And we know this because if you're hungry, for example, which is a different type of internal state, not an emotional internal state, but it's a different type of like sort of brain state, alcohol turns on some pathways and not others. And because it's very, very difficult to study emotion in animals, although people I think are making very clever behavioral ways to start to study this, we're starting to understand a little bit more about how emotional state might impact sort of how alcohol affects cells. Now, one of the ways that this could be happening is through early life stress pathways. And so early life stress in humans is one of the main predictors if someone is going to develop an alcohol or substance use disorder. And what early life stress can do is it can cause epigenetic changes to your stress pathways. And these epigenetic changes to your stress pathways can severely affect activity of your reward circuits, and also circuits that they think are important for, maintaining fear versus happiness, right? And alcohol can affect these pathways as well. So there's probably some really interesting interactions through which alcohol affects the epigenetic mechanisms caused by early life stress. But very few people are studying this right now. I would say give it 10 or 15 years and we may understand a lot more about how that happens. Part of it too is dose, right? You're very sensitive to dose of alcohol. And if you're happy or if you're sad, you might be drinking at slightly different rates and imbibing different amounts of alcohol. And different amounts of alcohol can affect the brain regions in different ways.
Okay, thank you. And another question. If flies that are used are not the same, could some of the flies used be more prone to be addicted to alcohol? And are there linkages in between addiction pronity and some inherited properties like eye color, wing shape, gender?
Wow, that is such a good question. In fact, that's the very question that John Hernandez in my lab is studying. And so what he's doing to understand this question, because all of the work that I talked about today, we were looking at sort of like flies at a one-time deal. But what he's doing, he's taking the same fly and he has them in an assay that in which the fly can control the dose of alcohol that it's getting. Yeah. And so as you put this fly in this little sort of like tunnel area, and then whenever it goes to one area, it gets a hit of alcohol. And it turns out that some of these flies like to spend a lot of time administering alcohol. And some of these flies don't like to spend very much time at all. They avoid the alcohol as much as possible. And if you put the same fly in this assay for 3 days in a row, those flies that start out liking alcohol actually escalate their use. And those flies that start out not liking alcohol very much can even reduce their alcohol use over those 3 days. And John has actually mapped this to one dopaminergic to acetylcholinergic brain circuit in the mushroom body. And so now he has an understanding of how sort of the aversive properties of through a dopamine neuron are affecting the appetitive properties through an acetylcholine neuron and how that's driving behavioral choice. So we know that there's one dopamine microcircuit involved in this sort of escalation. And now what he's doing are molecular experiments to figure out what's different in those flies that like to escalate their use or administer lots of alcohol compared to those flies that don't. And so the truth is we don't know yet, but we know that there's differences that are encoded into the circuits themselves. We don't know what the molecules are yet. There are other experiments that people have done in rodents where they've selected rodents for really long periods of time, and they've selected rodents that like alcohol and they don't like alcohol. And it turns out there's a ton of genetic differences between these different types of rodents. And so now they're just starting to map what some of these genetic differences are. So we know that there's differences that sort of last over many generations, but we also know that there's probably differences that are immediate. And it's just a matter of time, I think, before, our lab and other labs sort of map what all of these different types of things are and how they affect neural circuits.
That's that was just incredible. I'm checking for any if is there any other questions?
There's one that someone had chatted to me and they said, if alcohol changes our genes, instead, can we heal them by quitting alcohol? That's actually a really excellent question. And indeed, you can, because a lot of the gene expression changes that I talked about, they're not permanent. They require alcohol to be constantly imbibed. And if you stop drinking alcohol, like your gene expression can change. And even in those areas where maybe your gene expression for those particular genes don't change, you can impact other pathways, right? And those pathways can sort of compensate for the neural activity changes that are necessary to make strong behavior modifications. Yeah, and so nothing in the brain is stuck. Everything is plastic, and your behavior can actually change your gene expression. And so I think that's a really important question and point. Thank you for asking that.
Thank you for your answer. And we have another question here. Are there potential biomarkers at the molecular level that can help identify individuals at risk for developing alcohol addiction?
Ah, that's a really good question. I would say that there are no published biomarkers right now, but I know that there are new assistant professors that are identifying the biomarkers because we now have the technical capabilities to sequence human blood, like sequence the RNA in human blood, and then look to see in people with alcohol use disorder versus other, no alcohol use disorder or other addictions, if there are particular markers that can predict early stages. I would say, keep your eye out. Maybe in the next 10 years, we'll be able to see what some of those markers are.
Thank you. And we have another one. How does alcohol addiction alter synaptic plasticity and neurotransmitter release in the brain? Sorry.
So I would say that alcohol can affect synaptic transmission, synaptic plasticity, neurotransmitter release in very different ways. Part of it depends on the type of cell, and part of it depends on the dose of alcohol, and part of it depends on previous alcohol experience. So it can increase or decrease synaptic release depending on which type of cell it is and what's connected to that cell. And so in the cells that we're looking at, for example, we notice that alcohol will increase plasticity in dopamine cells, but it actually decreases plasticity in some of the glutamatergic cells that we're looking at and increases plasticity in some of the GABAergic cells that we're looking at. And so a lot of what we understand about this actually comes from slice physiology work in rodents. And so what they do is they take a mouse brain out, for example, or a rat brain, they slice it up and they put the preparation on, and then they'll diffuse very low concentrations of alcohol over the slice. And so a lot of the physiology that we understand comes from that preparation. It turns out that although we've learned incredible information on what alcohol can potentially do to a circuit, that isn't always how it acts in a living brain. And we're just starting to understand how alcohol is impacting plasticity in a behaving animal, like by using these sort of new genetic tools that allow us to understand how neurons are working and behaving in animals.
Thank you so much for your answer. And now I think we're done with the questions. And now, dear participants, I will now ask you to switch on your cameras for a photo shoot, please. If you can.
Yeah, it's pretty late there. I'd be in my pajamas already.
Your face, please. Oh, nobody? I'll take a picture. Or Everybody, please turn on your cameras.
I also added my lab website and my email to the chat, so feel free to contact me if you have any further questions.
Thank you so much.
There you go, a couple more faces.
We're seeing your faces.
Thank you all for joining me today, everybody.
So I'm taking the picture. Yeah, I think we're good. Okay, so first of all, thank you very much for joining us and presenting today, Dr. Karla Kaun. And it was an amazing presentation. And thank you for your answers to the questions. We're deeply proud and honored to have you here. As a Ankara University Neuroscience Society, we will be looking forward to meet with you all in our future events, everybody. Thank you all for attending our event. And don't forget to follow us on social media. Stay tuned. Good night.
Thank you, everybody, for joining me, and good luck with all of your studies.
Thank you so much. And again, good morning for you. Have a nice day.
Thank you. You too.